All right, so first of all, I should apologize to you because in the last lesson, I forgot the final part because it was on a separate PowerPoint presentation. But it's an important one. You might remember that I told you that not all animals develop tumors. So in that sense, humans are not necessarily a good guide to understand other animals and vice versa. And hence, tumors are not an inevitable consequence of being alive or being old. It's something that happens to humans and other animals, but not all of them. For that matter, plants also nearly don't have tumors. I said nearly because there is at least one instance of plant tumor, which is due to infection from a bacterium, Agrobacterium tumefaciens, which causes plants to develop, trees mostly, trees to develop bumps in their trunks. You might have seen trees with large bumps. The trunk goes like this, then you have a bump, and then it continues. So that's a tumor, yes. But it's a benign tumor. But in extreme cases, the plant doesn't die of it. And it's interesting, because in principle there should be no reason why animals, at least many of them, get tumors and plants don't. And so I remember that many years ago, a student asked this question to an oncologist who was giving a seminar, and he asked, why do animals get tumors and plants don't? And the poor guy didn't have an answer for that. And I understood that the student was repeating a question that somebody else had asked. And so there might be basis for that question. And I kept thinking about it. And I even talked to colleagues about that. But the issue is, oncologists don't know about plants, and botanists don't know about cancer. So neither part knows what's the difference between animals and plants. And finally, I think about 10 years later, that remained in the back of my mind. I kept thinking about it, and suddenly I got the answer. And it was surprisingly simple. And I think you can even think of it. So I'm not going to give you the answer. I want you to try and find it by yourself. And so the hint is, what is the feature that is inevitable in malignant tumors? What is it that a pathologist, the specialist in morbid anatomy, recognizes under the microscope and says, this must be a malignant tumor? You know? Think of the characteristics of tumors and think what is the most essential one in order to know.
Maybe the loss of cellular organization?
Loss of cellular organization, yes, oftentimes it is a good sign of a tumor being malignant, but it's not the hallmark that makes it malignant.
Come on guys, you know it.
I'm sure you do.
Well, I have an example for in humans, but I wouldn't know what the equivalent for plants would be.
Let's talk about
the
humans.
potential for angiogenesis, for example.
Angiogenesis, yes, it is necessary because tumors are tissues and need blood supply, but it's, again, not the hallmark that you're looking for. In fact, there are benign tumors that are very heavily vascularized.
Degrading the basement membrane, invading the cellular tissue.
Getting close. If you degrade your basement membrane, this applies only to epithelial tumors. If you degrade the basement membrane, what do you get?
You can reach the connective tissue and metastasize.
Right. Well, before metastasizing, the pathologist typically doesn't see the metastasis, doesn't even know oftentimes whether there are metastases. And yet, he unequivocally recognizes that it is a malignant tumor because he sees that the basal membrane has been broken, and after that, the tumor cells, what do? After breaking the basal membrane?
And the answer is not migrate towards the connective tissue.
Yes, they migrate, but not only migrate,
Invade
they invade the nearby tissues. That's the hallmark of a malignant tumor. They invade nearby tissues. Eventually, then they will metastasize. So, it came to my mind that plant cells cannot invade. In fact, they cannot even move because they have a wall around them. So, at the most, they can create a benign tumor which grows only by successive juxtaposition of numerous layers of new cells, which is exactly how our benign tumors grow. And so that's the difference between animals and plants. Yeah.
Are plants not able to produce enzymes to degrade the cell wall?
No, they don't do that. And anyway, they don't move. That's why, by the way, they don't have a central nervous system. They don't have any nervous system at all. Because the nervous system, first and most important, is needed to coordinate movement. If you don't move, you don't need a nervous system. So, why am I talking about that? Because last time we talked about aging. We talked about senescence because we were talking about telomeres, and then we switch it, or we passed inadvertently into aging because senescence and aging are correlated, as I tried to explain. And so there is another thing that characterizes humans in terms of aging. And I would like to show you an amazing review paper that came out several years ago. And I was astounded when I read it. For 2 days I couldn't think of anything else. So, first of all, let's say that senescence or biological aging is the gradual deterioration of function characteristic of most complex life forms. What does it mean? The older we get, the more efficient, the less efficient, the less fit, the less adapted to our environment we are. Our organs, our systems decay up to the point where they can no longer sustain life and we die. That's senescence. But as I mentioned last time, we don't really know the deep mechanisms that provoke senescence. So we are at a loss in determining mechanisms of senescence. And so we have devised indices of senescence, things we can measure and determine whether or not a given organism is of old age or not. What are these indices? Here they are. Mortality and fertility. Why mortality and fertility? Think of humans again. The older we are, the more likely we are to die in the next whatever interval of time, 1 year, 10 years. 10 years. So if you are 10 years old, you are unlikely to die in the next year. If you are 80 years old, you are much more likely to die in the next year. That's mortality. What about fertility? Well, you know that in young age you are fertile, and that fertility declines with age down to 0 at some point. So these are clearly markers and critical aspects of aging, or so it seems. Now, this is the paper I read diversity of aging across the tree of life. So these guys, these authors, considered the whole literature, ecological literature. They read all available papers in ecology and selected the ones that at the same time, with sound methodology, with reliable data, describe both mortality and fertility for whatever species they could find. And then they put together the data in a table. And first of all, I'd like to show you how the table is constructed. Each frame in the table is constructed. Here is the part of the table that concerns humans. First of all, they consider the present-day humans. What they have on the abscissa here is the beginning of fertility, so for humans about 15 years old. And then here you have an extreme old age, even though the most extreme ever recorded. And you have three curves on these graphs. One is fertility. Now, in this table, for humans, and only humans, fertility is considered from the female viewpoint. That's because it's easy to determine when a woman is fertile or not. While for men, it's indefinite, and most old men don't want to have children. So we don't really know when fertility ends, and it's highly variable anyway. So this is for women. Women become infertile nearly suddenly at about 52 years old. First curve. The second curve is mortality. It's nearly flat in the early years and then goes up literally exponentially. Finally, this is the how the population evolves. So you start from whatever number, say you take a cohort of 1,000 individuals, and then you wait until they have all died. So most of them remain alive for a long time, and then they all die within a relatively short time frame. Why is it? Because we live in a very protected environment. So we live as long as we can. We are protected from predators. We are protected from most diseases. And we try to protect ourselves from accidents. So we have few opportunities to die, fortunately enough. Then the biological limitations come and you die because you are too old, you are aged. Okay, this is modern humans. Japanese are one of the human populations with the longest lifespan. All right, so but the authors thought, well, maybe this is a very special condition, very modern life. Let's consider Swedish people born in 1881. In Sweden, they have very accurate records of the whole population from the 19th century. And so they looked at the data, and basically you see the curves are the same. Of course, fertility is not very different from this. Mortality has the same shape. They died at an earlier age, 89 rather than 102. But other than that, the shape of the curve is exactly the same. It's parallel to this. And the population curve is similar to the previous one. Some of them died earlier in life. They were less protected than the other people. But then again, this is not perhaps the natural condition in which humans evolved. So they considered a special population of hunter-gatherers, people that don't farm, that don't grow plants or animals. They just hunt animals and gather whatever they can find, whatever edible they can find around. But then again, things don't really change. Fertility is the same, and the mortality curve has has the same shape, although of course they died earlier still. Okay, so you now understand, first, how the graphs are constructed. Second, that humans basically behave the way these graphs show. Now, let's consider humans in the context of the whole tree of life, at least the part of it that the authors could find data for. Here is a complex table that I'll have you go through. First of all, where are the three human graphs we consider? 1, Japanese, 2, Swedish, 3, hunter-gatherers. All right. So, what about the rest of the table? The rest of the table contains a variety of different species in different kingdoms, and they have humans. They are color-coded. Humans are light gray. Other mammals are light brown. Other vertebrates are darker brown. Invertebrates are reddish. Trees are green. Other plants light green. Algae blue. All right. So, and how is the table organized? It goes from upper left to lower right according to mortality curves. So if you look up here, the animals in this part behave much like humans. There's a limited time for fertility, and each species is different, of course. But again, the mortality curves go up quite fast, here 2 on the second row. And you might say that, for example, this animal here, let's see what it is. Oops, no, it shouldn't have done this. Ah. It shouldn't. All right. For some reason there it doesn't work. There it doesn't work. Okay. Never mind. Let's see another example. Here, lions. You see that lions here, a very steep mortality curve, and they remain somewhat fertile for most of their life. Okay, but let's go a little bit down, such as here. Look at these curves. First of all, mortality climbs just a little at the very end of life. Fertility basically stays the same for most of it. It declines just a little at the end of life. Now, here, ecologists realize that these animals are swans. While studying them, ecologists realized that they didn't behave as expected. They didn't really increase their mortality rate. They didn't decrease their fertility. And so they made an extra effort to study very, very old animals. And eventually, with this exceptional effort, they found a little bit of increase in the mortality curve and a little bit of decrease in the fertility curve. But it was really minimal. And then if you go farther down the curve, things really change seriously. Look at this, for example. Here we have basically flat curves for both conditions. And this is a hook. Now, let's consider what it means that a hawk here has a basically flat mortality curve. At any age, it has the same probability to die. Think of it. It is as if a human 90-year-old human had the same probability to die than a 10-year-old boy. Or girl. That's surprising. Yes.
When we consider mortality, we consider like death from natural causes or also like from other predators?
From any causes, including diseases, predators, and accidents. So eventually, even if you, let's assume for the sake of the argument that you are immortal, it doesn't mean that you will live forever, because sooner or later you will have an accident, you will have a deadly disease, or you will be eaten by a predator. Right? That's why these animals die, even though their mortality curve doesn't change. But if their mortality curve doesn't change, it means that they remain fit. Even when they're old in their lifespan, they are as fit as when they were young. So they are not aging. Yes?
I didn't realize we were also including death from not natural causes in mortality. Because if we do, I don't understand how the mortality is comparable in hunters and gatherers and Homo sapiens, like the Japanese would do.
It is comparable in the sense of the shape of the curve. But you see that the curve here is much shorter than because they die earlier. Because they are, yeah?
Why isn't it also steeper? Like going up in a higher rate?
Let me think. It shouldn't be. It shouldn't be. You simply restrict the actual lifespans. Here, see, here you see it's up to 81, here up to 89, here up to 102.
So if we had the same measures, it would look steeper as well. But
Exactly.
we have,
Exactly. Because you have higher chances of dying by accidents rather than non-natural causes, then your lifespan is short. All right, let's go on. And let's, you see here, most of the curves are flat. And I need to emphasize that, for example, in this case, fertility increases with lifespan compound. And this is a blank. Let's look at this particular case. This is an interesting one. This is Hydra. Hydra is a very small, 2-3 millimeters long animal that lives in water. And the data here come from an experimental work, a heroic experimental work carried out by the authors of this review, in which they followed, actually there were 2 independent laboratories. Each one of them followed large cohorts of hydras kept in the laboratory, cultivated in the laboratory, for 20 years. And they recorded each death in these hydras. They had 1 animal per each well in multi-well plates. That's the familiar setup in the laboratory. So whenever an animal died in one of these wells, they recorded it. They recorded all causes of death, including accidental death, like dropping a plate to the floor. So, despite that, the death rate was extremely low, so low that they extrapolated. Of course, they couldn't follow the whole lifespan of these animals, because by extrapolation they found that they lived at least 14 centuries, 1400 years. And these are very teeny, very delicate animals, most of which die during summer, simply because the pools of water they live in dry up. So evolutionarily, it doesn't make sense that an animal that is destined to die very soon will develop the ability to survive basically forever. And so that's another very surprising result. So, and again, the fertility remains absolutely low. And you have similar situations in these other ones. Let's look at these. Out of curiosity, what else? This is a living plant. This is mollusk. This is a frog. Good. Also, yeah.
In the case of the hydras, you said that they have to.
In the case of the-
In the case of hydras, you said that they have to extrapolate like the rest of the lifestyle. What's the oldest recorded hydra that they have in the world? Are they able to study how old is that particular hydra?
They cannot tell how old is an animal. So they start from a point which is arbitrary and they go on for 20 years. Okay, now let's look. Oh, by the way, I forgot to draw your attention to the population curve. You see the shape it has in humans and the upper rows. It's basically a convex curve. Look here. It's a diagonal curve. It's a line. What does it mean? It means, again, that the death rate remains the same throughout life. Slowly, one by one, the animals or whatever living being they are die, and eventually the whole cohort is gone. But again, according to our criteria, they remain young until their death.
And that line becomes the more diagonal, the more the mortality decreases.
Yes. To the point that if you look at the lower row, it is no longer convex, it's concave, because with these species, mortality actually decreases with age, which is most amazing. And fertility increases. And so we want to look at all of them, just to make sure that we are not looking at exceptional, very strange cases. This is coral. This is a flowering plant. This is an algae. This is an oak. This is tortoise. In fact, it is one of the longest living animals. Actually, the longest living animal we know of. And the final one is mangrove tree. All right, so I think I've explained already that this doesn't fit with our preconceptions about aging. Whence the conclusions of the authors? Let's see what they are. Maybe aging is not universal in biology after all. Some species we have considered don't seem to age at all. Scientists must gather more and more accurate empirical data on mortality and reproduction in a much wider range of species. The data are insufficient to draw firm and widespread conclusions. Furthermore, and this is conceptually relevant for would-be physicians like you. Oh no, this is another conclusion. Theoreticians, I told you that aging is a matter of, it's largely approached by theorizing. Theoreticians must devise theories that account for negligible or negative senescence. Negative senescence is what you've seen at the bottom of the paper. And finally, for you, in such empirical and theoretical studies, researchers should guard against anthropocentric intuition about aging. Humans, especially modern humans, are extreme outliers. So we are exceptional animals, and not in a good way for that matter. Most of us probably would like to live for a longer time, potentially forever, but we don't. We are not. As we oftentimes think or have an intuition of, we believe we are at the top of the animal kingdom, we are the best animals in the world. No, it's not so. We have the best brains in the world, probably. We could argue that. But by other, according to other parameters, we are rather poor. For example, in walking and running, we're pretty slow. But most animals, large animals, walk and run much faster than we do. Yes, we have hands, of course. That helps a lot. We wouldn't be here if we didn't have hands. But we pay a price for it. And so we pay a price for other characteristics of our bodies, which entail that we have tumors, that we don't live as long as others, that we age dramatically throughout our life. So we are not typical animals. We are extreme outliers. So this is the part that should have concluded the previous lesson. Are there any questions about this before we go on? Yes.
Yes, maybe it's a bit too general, but would it be able to pinpoint which point in difference in evolution takes away that ability or increases the rates?
At this time, there are absolutely not enough data to go that kind of
Hypothetically,
hypotheticals. That's a difficult question, but I can point out something. First of all, in this table, species are rather mixed. You know, there is no prevalence of whatever kind of living beings anywhere in the table. But animals tend to be more represented in the upper part of the table, and plants in the lower part, brown versus green. So there is a tendency for plants to grow, to live longer than animals, and to remain fertile longer than animals. Also, simpler animals seem to have the ability, they have two abilities that at some point, at some level, probably are correlated. They have the ability to live for a very long time, and they have regenerative abilities. We don't know exactly how these two abilities are correlated, but we suspect that they are at some level. And then if you go up, let's say we call it up because we pretend to be at the top. If you go up in the animal kingdom, let's restrict our argument to be within the animals. If we grow up, you tend to lose both the shape, the flat shape of the mortality curve, and the ability to regenerate, with exception. It's not a linear relationship between complexity and those features. So, right now my guess would be that the more complex living beings become, the more difficult it is to maintain the highly organized body plan of theirs. And so they age, as we said in the last lesson, for physical reasons, because they cannot keep their entropy low, because they are too complex to remain organized. But that's a guess. It is not something based on hard science. Yes?
So you said that, like in the lower parts of all these graphs, they have quite like a flat mortality curve, but their lifespan doesn't seem to be that big anyway. So what's the point?
That's, well, as I mentioned, that depends on, as I said, even if you are immortal, you die eventually, for some reason. So their actual lifespan depends on their ecological niche. If their ecological niche includes many predators, many chances of being killed by accident, many diseases, that is, the possibility of getting deadly diseases, then they will live long anyway. Think of plants. Just a drought will kill them all. Right? So, the surprises are not finished here. We just switched to a different topic. So let me do it. The current topic is this. Human genome modifications, which means enhancing humans, with a question mark. Can we do it, and should we do it? That's a fascinating topic, which, as I mentioned last time, stems from a question that a colleague of yours asked at the end of my first lesson last year. She asked, if it is true that some animals, such as mules or elephants, don't get tumors because they have extra copies of PP3, can we have extra copies of PP3 in humans and make them resistant to tumors as well? And the answer was yes, we can do it. And she said, have we done it? No. Why haven't we done it yet? Because we have chosen not to do it. And so that was sort of a surprising answer on my side, or was perceived as such, because nearly everybody in the class thought, why don't we do such a good thing? Why are we not doing it already? And so this lesson stems from that question and tries to explain what we can do, why we might want to do it, and why we shouldn't do it. And then eventually I will ask your opinion on this. Well, eventually, as mentioned also during the lesson. So, can we generate humans that are more resistant to tumors, that are better than we are, in some sense or another? The answer is probably yes. How? By gene therapy. Meaning, in this case, not therapy of an existing disease, but a preemptive therapy aimed at preventing disease or other conditions that we don't like. So, gene therapy can be subdivided into two classes. Gene therapy of somatic cells and of germ cells. Now, I remind you that somatic cells are all the cells of our body except those whose task is to generate a protein. All right, so with somatic cells, we have no problems. We are already doing gene therapy of somatic cells. The reason why we don't have a problem is because when we do that kind of therapy, we only cure, in the best case, of course, when we succeed, we only cure an individual. That individual is cured of its disease, but that progeny is not. Because our germ cells still bear some genetic alteration that can be transmitted to the progeny. With germ cells, you can in fact eradicate completely whatever genetic defect given family carries and eliminate the genetic disease from that family. So, the main purpose of germ cell gene therapy is correction of genetic defects. How do we do that? There are basically two classes of interventions that we can perform. The simplest is transgenesis. Transgenesis involves taking a gene that we like, that we want our patient to have in his cells, and integrating this gene into a random locus in the cell's genome. Basically, what we do is simply we introduce this piece of DNA into the cells of the patient, and then we wait until those cells integrate into their own genome this piece. And so, in each cell it ends up in a different place, which is a problem. But anyway, this method has been applied in human gene therapy, and it works. There have been some provoked tumors, by the way, and some deaths, acute deaths, due to gene therapy. So it's not in our case. But it can be done. There is a subtler, better way to do gene therapy. And that is to do genetic recombination. This is what we call, this is the final outcome of transgenesis, by the way. You have inserted this gene into a chromosome, and it now stays there forever, and it's carried down to the progeny of the cell. On the other hand, we have a more sophisticated technique called, by the way, the method for transgenesis got a Nobel Prize, and this other method got another Nobel Prize, just to assert the relevant importance of it. Suppose you are in a situation in which your patient has a mutation in one of these genes. With transgenesis, and this gene doesn't work. That's the problem. He has a disease because one of his critical genes doesn't work. So, with transgenesis, you might insert an extra copy of this gene with a wild type sequence and replace this one. But as I mentioned, there are problems with transgenesis. The other option is to correct the problem, to correct the sequence of DNA here, which looks like magic, but it isn't. You do it this way. You take a copy of the same region of DNA in which the mutation is, but now put the right sequence here. Then you allow the cell to recombine your exogenous DNA with the endogenous one. Recombine means the cell recognizes that the two pieces are nearly identical and exchanges them. So it inserts this not within the genome, but in place of the original genome. Is that clear? So you're basically replacing the gene, or whatever region of the gene you want to modify. And so the final outcome is that you have the right sequence in the right gene, in the right place in the genome. You have reconstituted normality. You are perplexed.
No, I was thinking of something else.
That's a lot. All right. Practically, how do you do that? You take an embryo stem cell. In the case of humans, if you wanted to do it, it would have to be a disrupted embryo from which we take a cell. Or we have the ability of taking a very young embryo, take one or a few cells from it, keep it alive, implant it into the uterus, and so it will grow. So we can take the very cells of the embryo we are trying to manipulate. This is commonly done in animals, in mice, and other animals. We modify these embryo stem cells genetically in the way I've shown you, and then we implant the modified cells into the blastocyst, which is again one stage of the embryo, and they become part of the embryo. So at least some of the cells of the newly generated organism that we develop from this embryo will carry the genetic modification that you have performed. Here is a microscopic image of the procedure. Nowadays, this is very recent, we've been able to do something better. We can actually take a zygote, a just fertilized oocyte, modify it genetically, and then implant it. Why is it better? Because this procedure here generates a chimera. The animal that comes out of this embryo has some original cells and some modified cells. And they randomly place themselves all over the body. So you cannot predict exactly where the genetically modified cells will end up. If you are unlucky, they will not go to the germ cell line, and the genetic modification will not be transmittable. If you directly modify genetically the zygote itself, then the whole animal will be genetically modified in the way you like. All right, so I wanted to explain a little bit the technicalities of what we do. And I emphasize that we do this routinely with animals, and there is in principle no reason why it shouldn't work with humans. And in fact, this is a spoiler, it has worked with humans. So, this is, I've called it gene therapy of the germline. In principle, it's highly desirable. We wipe out a genetic disease from a family. That's very nice. It is certainly a desirable goal. Problem is that this technique is complex, costly, dangerous, and it's not really necessary to fight genetic disease, because this is a report of the American Association for the Advancement of Science. So many scientists thought about the problem of gene therapy of genetic disease at the germ cell level, and they concluded that we don't need gene therapy for that, because we already have tools that can attain the same goal, eliminating the genetic modification, wanted genetic modification of one family. These tools are genetic counseling. Don't marry your cousin, in short. Artificial heterologous insemination, prenatal diagnosis, therapeutic abortion. Now, I know that there are ethical concerns about some of these practices, but they are part of modern medicine. And so, with a combination of them, we can basically, in principle, we could eradicate genetic diseases at a fraction of the cost, of the time, of the suffering that gene therapy of the germline would cause. All right. Nonetheless, several years ago, this paper came out. It was 2001. This was a scientific journalistic paper relating the scientific fact. What was the scientific fact? They had created the first monkey, genetically modified monkey. Now, monkeys, of course, are close to humans. That's why it's relevant. This guy here is called Andy, which is the inverted acronym of inserted DNA. It was supposed to bear a GFP gene, which fluoresces under UV light, under ultraviolet light. So this monkey here was supposed to become green if you shone it with an ultraviolet light. It didn't. So basically the experiment failed, but it was the first genetically modified monkey. Yet the paper was entitled Biology's Last Taboo. Why was it? Because we have said, well, probably we don't need gene therapy of the germline, but anyway, it aims for desirable goals. So why should it be a taboo? The reason is because you can use gene therapy of the germline not only to prevent genetic defects, but also to improve humans' characteristics. And this is shady. Basically, we could use it to transcend the limitations of our biology. That's a very interesting perspective to consider. But anyway, again, biology is immensely complex. Our bodies are immensely complex. Look at this. This is an interaction map of all the proteins, or many proteins, that make up the body of a C. elegans, Caenorhabditis elegans, which is a very teeny worm made of little more than 1,000 cells, really a very small animal. Yet its proteins, and thus its genes, interact in such a complex network that basically, if you touch any of these genes or any of these proteins, you are nearly assured to modify many, many other parts of these networks, even distant parts, because they're all connected directly or indirectly. And so, the question you might ask is, what are the chances that an apprentice watchmaker, such as we are, with respect to biology, by adding a cog or a spring into a precision watch, would improve it rather than break it? No. Sorry?
Very little.
Very little, very small, nearly nothing. Right. And yet, amazingly, it seems that we can do it. The reason probably is because nature never achieves perfection. Nature achieves organisms that are good enough to survive in the environment they live in. In principle, we can, why are you laughing? Are you also thinking of something else? In principle, we could improve on organisms that have been selected by nature. Because nature doesn't aim to perfection, nature doesn't aim to anything, doesn't have any intention. This is important. There was a colleague of yours last time that asked me a number of why questions. Why are, no, it was a woman here. Why questions of the kind, why has nature done this? Why are these animals like this? But you cannot ask why questions in biology. Why implies intentionality. Why have you done that? Because I wanted to achieve that. But nature doesn't have intention, doesn't have purpose, doesn't have aims. So you cannot ask why questions. You can, at the most, sometimes ask why questions for shortness. When I ask why, I don't know, we have nails, why do we have nails? I really mean, what is the selective pressure that has made so that people with nails survive better than people without nails? So I shorten this long question and I ask, why do we have nails? But that's an incorrect way. I do that for shortness, but we should all understand that one question must be considered with regard to evolution. Evolution within, I've seen your hand, I'll come to you in a second. Evolution within the constraints that evolution itself imposes. That is, I've mentioned, we had only 2 legs, which makes us slower. That's no good. If a lion runs, or a bear runs to you, you have no chance to escape. Not by running. Choose another way. Why do we have only 2 legs? Because nature found useful, we agree with that, having arms with hands. We are humans because we have hands before having our legs. But wouldn't that be been better to have 4 legs plus 2 arms with hands? Would have been better. But nature is unable to do that. There are no mammals like that. Because the body plan is fixed. You cannot modify it by any single random mutation. It's extremely difficult, nearly impossible. In fact, it has never happened that a mammal was developed with 6 limbs. So there are constraints to what evolution can achieve. He first, then me.
I was thinking, when you phrase it like that, which I completely agree with, nature not having a purpose, but just functioning evolution-wise. The analogy with a watchmaker stops being extremely accurate, and the potential of the apprentice changing something to make the watch better becomes higher. Because in that case, the watchmaker designs it to be as perfect as possible. Whereas here we're working kind of differently.
Exactly. That's what I was arguing. That in fact, even an apprentice watchmaker can improve a watch because it has an aim. And the watch is not perfect. It's just the best people have been able to make so far.
And it's kind of connected to what he said. Since nature is, let's say, random in this case, and you said that we get legs because we have hands at this point, but then monkeys have 4 hands and 4 legs at the same time, so they get that advantage. They're stronger than us.
Okay, so that's a bit complex, and I'm not a zoologist, so let me try to answer that. Yes, monkeys have 4 legs and 4 sort of hands. They are probably superior to us in terms of limbs. They don't have our brains that allow us to use our hands in a way they are not. We are much finer in controlling our hands than they are. And so we are able to produce things like this, which is very simple in principle, but unless you have very fine hands, you cannot make it. Simply. An animal would never be able to make this.
Like several times during several courses that we had, we would say that like having too many genes would increase the genetic burden. So at which point does like adding more genes in our genome becomes dangerous? Like the cells can't handle that much DNA.
Well, the first answer is that one or a few genes, more or less, doesn't really matter. We have about 20,000 genes, so a few more or less won't change the general picture. Second, yes, true, the more genes you have, the higher the chance of them, the larger, the wider the target for mutations to hit. But basically, having our number of genes, it is not the most genes that animals have, but close to the most. There are animals that have like 100,000 genes. Not that I know of. There are plants that have many, many more genes than we do. But that's because among plants, genome duplication is very common. It happens in animals, but it's much rarer. So many plants are the results of whole genome duplication. So they double all of a sudden the number of genes they carry. And then the genes, because they are in multiple copies, could be modified. You can modify one copy while keeping this other one in its original form, so that you have both functions. So, can we actually modify genomes that nature has selected over millions of years and make them better? First of all, I want to emphasize that making an animal better must be, an animal can be better only with regard to the environment in which it lives. That is, some genetic traits might improve us in this particular environment, but may be detrimental in another environment. Classical example is, suppose we have a genetic modification that allows us to use up more calories than generally we do. So the obesity problems that we have in the West will be eliminated. But what if a famine comes? Then a desirable trait today will become a deadly disaster in the future. So it depends on the environment. There is no perfect biological being. There is only a biological being adapted to the environment in which it lives.
So you don't think there is an objectively advantageous trait?
No, I don't think so. It really depends on the environment. I think you can, I've never done this exercise myself, but I believe that you can always think of an environment which would make any trait undesirable.
Not even a couple of deficiency genes?
Yes, I can tell you what. And I will in a few minutes. So, let's see what we can do with mice, and then let's talk about what we might be doing with humans. Okay, look at these mice to start with. We'll start softly. This mouse here is a wild-type mouse. This is a transgenic mouse that overexpresses IGF-1, insulin-like growth factor 1 receptor. No, sorry, it's not a receptor, it's the growth factor itself in the skeletal muscle. You immediately see that this mouse is bigger than its wild-type leader mate. They're brothers, in fact. But this guy is wild-type, this is transgenic. It is bigger because its muscles are much bigger. And so you might wonder, well, in our current environment, muscles are not that important. Many of us spend time in the gym, right? We don't need strength, muscular strength. We have machines, we have our environment that prevents us from carrying out many tasks requiring physical strength. So muscles are not so important, except that these muscles look at the difference, by the way. They look like bodybuilders. But these mice remain strong even in advanced and very young age. That's important. It is important not to develop muscle waste, which we do, many of us do in old age, being unable to attend to ourselves. It would be nice to remain strong in old age. So that's appears to be a desirable trait. Now, it is important to notice that these animals here apparently have no evident abnormality. They live as long as the wild type animals. They're fine, they're fertile, they're apparently happy. So they don't pay a price for their increased strength. Except they have to eat more to maintain these muscles. Now, no.
Do their organs also grow in size?
No. It's just this is because overexpression of IGF-1 is confined to skeletal muscles. A generalized expression will be toxic. All right, so another interesting example. We talked about, very pertinent to the course here, we talked about tumors. So can we make mice resistant to tumors? Yes, we can. And this is of course desirable from our point of view, and it's interesting because it answers your question as to whether an extra copy of PP3 would be desirable. First of all, let's see what happens in mice carrying one extra copy of PP3. First of all, they have many fewer spontaneous tumors. Mice develop spontaneous tumors with the same frequency as we do. But these PP3 mice develop many fewer tumors just because they have an extra copy of PP3. But even more strikingly, they are resistant to tumorigenic chemicals that induce tumors actively. This is a histology of skin of mice treated with methylcholine, which is a very well-known carcinogen that you can put on the skin, and the skin will transform, will produce papillomas and eventually carcinomas, and the mice will die of these carcinomas. These are wild-type mice. They are basically all dead by 40 weeks after treatment, and these are the PP3 mice. The majority of them is still alive at the same time. From the ethical point of view, we don't allow the animals to die of the tumor. We kill them without making them suffer when the tumor is large enough that they will surely die. All right, this is another example. This is a urinary bladder treated with aromatic amines, very dangerous tumorigenic substances that smokers accumulate in the urines, and urines, of course, accumulate in the urinary bladder. And so these amines produce papillomas and then carcinomas. Here is the difference between wild-type mice. This is again mortality. They're all dead by 40 years after the initiation of mine treatment. And this is PP3 mice. Now, this difference looks a little less striking. There is a difference, obviously, it is statistically significant, but it's less striking than this, except that normal mice develop multiple papillomas in their bladders, and these mice mostly have one, which would be much easier to cure surgically in case you think of carrying this extra copy of PP3 into a human. Is an extra copy of PP3 the only way to prevent tumors in humans? No, it isn't. Sorry, in mice. It isn't. For example, you can add an extra copy of a particular genetic locus, the INC4ARF genetic locus. It takes the names of two genes because they are superimposed on one another, on each other, and but on a different reading frame. So they are entirely different from each other, and yet they overlap. Okay, so here we have mice. These are not wild-type mice. These are mice in which the INC4AR locus has been deleted homozygously. They don't have it at all. So they develop tumors fast. By the age of 70 weeks, they are all dead. Normally, a mouse lives about 100 weeks. Now, this is what you get with a single copy of the locus. So a heterozygous deletion of the locus. They live much longer. They still develop tumors more than natural than wild-type animals, but they do. But then if you replace it, you take an animal without the locus, and you add a transgenic locus, you approach the same situation as the heterozygous. These is spontaneous tumors, and these are lymphomas, sarcomas, and plasmacytomas, all blood tumors that develop in response to chemicals. Anthracene, and Pristin. So again, you can use genetics to prevent tumor formation, even in the phase of carcinogens. Now, I was going to answer your previous question. Please ask me one.
Is there a significant difference between the heterozygous mice and the transgenic mice? Is this slightly more?
I don't know, it is not shown here, but let's say there is. Probably because the transgene doesn't work as well as the natural ones. Going back to p53, we are asking, is there a problem in having an extra copy of p53? Well, yes and no. This experiment, to my knowledge, making mice with an extra copy of p53 has been done 3 times. In 2 of them, the extra copy was introduced into the mice as a transgene. That is, an artificial piece of DNA made in the laboratory and then inserted into the genome of the mouse. This artificial gene had p53 under the control of a constitutive promoter, which means p53 was constantly expressed at the same levels. Now, 2 of these mice were made this way, and they were protected from tumors, but they lived less than their wild-type real mates. The reason is because p53 is also involved in cell senescence. And so the trade-off there was, yes, having p53 constantly on protects you from tumors, but you die earlier because you have more senescent cells than a regular mouse. However, yeah?
I'm sorry, what do they die from then?
Basically all diseases of old age.
They age faster.
They age faster, yes. But the 3rd time, the guy was smarter. He took the whole genomic region containing the p53 gene and introduced that into the mouse. Now, in this way, p53 was regulated in a physiological way. p53 is supposed to come up only in certain circumstances, not to stay constantly up. So when p53 was physiologically regulated in terms of transcription and translation, then the mice were protected from tumors and lived as long as their wild-type lived. So that's a potential trade-off, but it also shows the technological way to avoid it. And I think we can make an interval. All right, so we were at this point. We have shown the ability to largely prevent tumors, both spontaneous and induced in mice. What else can we do with mice? We can extend their lifespan, which is also extremely impressive. Here is the first example of genetically extended lifespan. It dates back to 1999, and it was discovered by accident. People were studying this particular splicing form of the Shick gene. The Shick gene has 3 alternative splicing forms. This is the largest, P66. And they wanted to know exactly what that form, not the others, did. So they devised a rather sophisticated, for the time, mouse, which did not express only that form, but expressed the other 2 forms of the Shick. And so they studied these mice extensively in order to find out what differences from wild-type animals knocking out this particular splicing form provoked. They couldn't find any. And they looked at difference everywhere, and they couldn't. And so eventually they lost interest in these animals and abandoned them until the story goes, I'm not sure if it's true, or sometimes legends go on, but the story goes like this. At some point comes the bill from the animal house, because housing animals costs. So they say, oh wow, let's kill them. We don't need them because we can't find anything with them. And so they went to kill the animals, and they found, to their amazement, that these animals were alive and their wild-type controls were all dead of old age. So they say, oh, there is something to it. And so they eventually developed these curves which show that wild-type animals live on average about 800 days, these particular cohorts. We measure it at half the population. About 800, the P66 Shick knockout mice with nearly 5 days, which means, if you translate that into humans, it would be as if a human lived 100 years on average, not in exceptional cases. That would be, I would say, a desirable goal. And evidence is that this thing would work with humans, though we are not sure, we cannot be sure, based only on our experiments. And as I said, these animals were so normal that they couldn't find any differences with normal animals. In fact, they found a difference later. Again, I'm not sure the story goes like this, but I heard it from a very scientist who developed these mice. At some point, they had a problem in their animal house. The heating system didn't work properly and the temperature went down. These animals died. The wild-type animals didn't survive. The reason is because this particular isoform is involved in generating heat from the energy in the form of heat. Unfortunately, while producing heat, which is good, because you need it if you are in a cold environment, they also generate free oxygen radicals, which produce DNA mutations. That's why these animals, the wild-type animals, which produce more free oxygen radicals than the knockout mice, live shorter lifespans, because they accumulate mutations more rapidly than human mice. This is a bright example of trade-off. In the basic environment, the mutation is innocuous, and in fact it is beneficial in terms of lifespan. If you are in a cold environment, it is clearly detrimental. So if you want it,
I didn't understand that. Why is it clearly detrimental? Aren't the wild-type mice with the B66 the ones that are producing more free oxygen radicals?
and more heat.
So, is it really a trade-off? Because both of them have the disadvantage in the cold conditions.
No,
The normal
producing heat is good in cold conditions. You have to keep your body warm.
but you have the disadvantage of the free oxygen radicals.
Yes.
Whereas in the normal conditions, one
No,
on
sorry, no. There is a misunderstanding. Wild-type animals produce more heat, and more free oxygen radicals. So they're protected from cold, but they accumulate more mutations. These animals accumulate fewer mutations, but are not
Other prone to cold. But if I can even say that, if we manage to get an average out of the situation in non-cold conditions, let's call them normal conditions, the knockout mice have advantages and not disadvantages.
Yes.
And in cold conditions, both have advantage and disadvantage.
You could say so, yes.
So would it be unfair to say that in an average, it's better to be the knockout mice?
It depends. If you live in
On the
Siberia,
conditions, yeah.
no, it is a better.
So they die instantly in the conditions of cold.
Well, not instantly, but they cannot keep
Sure.
their body temperature normal. So.
Okay. Okay.
All right. So, and then there were more and more examples of genetically induced extension of the lifespan. For example, here we have, this is something I participated myself in. A colleague in my institute produced transgenic mice that overexpress MTH1. MTH1 is an enzyme that removes oxidized nucleotides from the nucleotide pool. This is very important because if you have oxidized nucleotides in a nucleotide pool, when you synthesize, when you duplicate your DNA, you insert these oxidized nucleotides into the newly formed DNA, and those cause mutations. So you need to keep your pool free, or as free as possible, of oxidized nucleotides. And in fact, these animals do it. You can artificially induce nucleotide oxidation, and the transgenics overexpressing MTH1 are better at removing the oxidized nucleotides. So, because it is believed that aging is in part due to the accumulation of DNA mutations, I predicted that these mice would live longer than wild type. And in fact, they did. We did an experiment on purpose and measured a difference of about 15%. The lifespan was extended 15% compared to wild-type animals. And this, yeah.
Do they end up dying from the same causes just later, or from different causes completely?
Interesting point. This hasn't been studied systematically. We haven't noticed any major differences in the causes of death, but these mice have not been studied by systematic autopsies. So we are, as far as we can tell, there are at least no obvious trade-offs. It appears that these mice are perfectly happy. They just live longer. Or, remember last time I told you that senescent cells within tissues causes a variety of problems, including chronic inflammation, including a pro-tumorigenic environment, including defects in the functioning of those tissues and organs. So these are mice that express a caspase, death-inducing enzyme at the cellular level. Enzyme that induces cell death, caspase A, under the control of a gene expressed in senescent cells. Now, these are two mice of the same old age. This is the wild type, and this is the transgenic. So in these mice, senescent cells are killed by the expression of caspase A. And so they are relatively free of senescent cells. Look at the phenotypic difference of these mice. Whoever knows mice knows that this is an old mouse. Look at the fur, which is far less smooth than this animal. Look at the hump, which is characteristic of that. Look at the eyes. This guy here looks younger. It isn't. It is just as old as this one. But it is in better general shape, in better general conditions, simply because it continuously gets rid of the senescent cells that get generated along the way.
In the last lecture, we said that senescent cells are rarely found independently of the age. So why are we saying right now that the old mouse has more senescent cells?
Well, this mouse has more compared to this. It has the normal amount of cells found in a wild-type mouse.
Okay. And the other one has
The other one is fewer, much fewer. All right, what else do we have here? Similar mice that now do not only live longer, but they have fewer spontaneous tumors. Again, here you see males, death due to spontaneous tumor occurrence, and these are the transgenic. Females, sorry, what's the difference between these two? Yeah, these are both males and females. Oh, different background. This is a technical thing. If you take an inbred strain of mice which are identical in genetic terms, so you cross them, brothers with sisters, for 20 generations at least, and then you get a strain of identical mice, this case called black 6. These mice are all identical, and so in principle they might carry deleterious mutations. And so you are not really making them stronger with your intervention. You're just correcting those mutations. And so you control for this by using a mixture of different strains of mice to make sure that it is not, your results are not due to the particular so-called genetic background that you have chosen. This is, for curious people, it is not necessary to understand what I'm saying. All right, so let's summarize what we have said so far. We can make the animals stronger and remain strong in old age. We can make resist tumors. We can make resist tumorigenic substances. We can make them healthier in old age. What else would we desire? What else would you desire for your children? It is too late for you, forget it. But what about your children? What else would you like to have? You can do even better than this. This is the Doogie mouse. The name comes from a cartoon I've never seen, so I don't know. That's the original. But this Doogie mouse is another expression of the NRB2 subunit of the NMDA receptor. This looks very complicated, but basically it has another expression of part of a receptor for a neuromediator. And it has an amazing memory. It is much, much better than wild-type litter needs. For example, in tests like this, finding a hidden platform that is beneath water, opaque water, they cannot see underneath. And so they swim until they casually find the platform and they finally can rest. Wild-type mice, when repeatedly challenged in this setting, don't quickly remember where is the platform. And they swim randomly again until they finally record in their memory where it is and they swim directly to it. Doogie does it immediately. After finding the platform the first time, he still remembers. And his memory is so good that he performs so well in such a large number of tests that people have argued that it is in fact it has not only a better memory, it has a better overall intelligence. It behaves as if it were more intelligent than wild-type humans. Wouldn't you like your children to be much more intelligent than the average human being? Probably you would. All right. So, yeah.
Could you explain a bit more what the NMDA receptor is? Or what does it do?
Oh, sorry. So, as I said, NMDA is a neuromediator. So, a number of neurons, not all of them, but a number of neurons have specific receptors for this neuromediator. And for reasons that frankly I don't know, and I think even the authors of this work don't know exactly. They just suppose that modifying this receptor will remember the anything was true, but I don't think it is known exactly why.
Maybe because the receptor may be involved in long-term potentiation, which is an important function of the process.
Very possible.
Wouldn't the hyperstimulation of these NMDA receptors cause the brain to consume even more energy? Because we're building.
I don't know. I don't think anybody has measured it, but it is possible. The brain already consumes a lot of energy, so that might be one trade-off. There might be other trade-offs. So having a larger, stronger memory in an otherwise normal brain might unbalance the delicate equilibrium of our abilities. And this is a particularly difficult example because we can physically examine the other animals that I've shown you that are stronger or more resistant to this or that. We can physically determine whether they are well, whether they live long enough, etc., etc. But you cannot ask Doogie if he is well, if he feels well. And so carrying these mutations into humans will be particularly debatable. On the other hand, fairly recently I came across a paper describing human beings naturally endowed with immense autobiographic memory. So here, these people remember every day of their lives, in all details. You can ask them, what did you do on Wednesday, April 12, 1984, and they will tell you exactly. It's amazing, but it is so. I even interviewed in depth a colleague of mine who was involved in this paper, a psychologist, and it was very interesting because he told me some things that weren't published. They examined these people psychologically in detail, and they found no abnormalities. They had a significant deviations of their compulsion. They tended to be extremely accurate in doing things. But that was still within the normal range, within the differences that we would find among us. So they were normal psychologically. They just had this huge, immense memory. So even doing this with humans might not be dangerous, although it is impossible to determine until we do it.
Were those individuals scattered across the population, or were they like localized to certain areas, or like
No.
genetically?
They're extremely rare individuals that scattered all over. It is similar to, there is another kind, at least another kind of superhuman memory that is called eidetic memory, whereby the person who has it remembers every detail that he sees, which means he looks outside and then he tells you how many windows, trees, cars, people, and things he's seeing, and he can even draw a very faithful drawing of what he sees. I've seen an amazing example of a person who was, sorry?
In a TV series, we have a character.
Exactly.
Sheldon.
Exactly. In this example, one of these people was flown on a helicopter around Rome, all over Rome, and then he redrew the whole of the city on a very large drawing. And every building was in the right place, every building had the right number of windows, in every possible detail. It's amazing. And this is a digression now, but I think it is interesting. I asked my psychologist colleague, how is it possible? Where do they put all these memories in their brains? How can they do that? And his answer was surprising. He had no proof of what he was saying. But he said, I think we all do that, except we normally cannot retrieve those memories. They can do it. In fact, I can measure that those people, they still need some time to retrieve a memory. If you ask, what have you done on that day, they think a little and then they tell you. But they think for a shorter time than you do for the same kind of task. And so, yeah.
So don't you think it is due to stimulation? Let's say people from rich, what would I say, like a boy from a rich family doesn't need to think like what is the alternative, but a guy from a poor family has to think other possibilities. Maybe overstimulating brain makes them better than a person who does not use the brain?
Not likely, because they show their abilities when they're very, very young. So they seem to be born that way. And so what my colleague was saying, I think we all store all of those memories, we simply cannot retrieve them. And I said, but that's a waste. A waste of space, a waste of room in our brains, whether we waste all this energy. And he said, he drew a very interesting parallel. I think, he said, it's like the hard disk in your computer. You don't get rid of all the files you don't need, because it would be an immense task. You spend hours and hours determining whether you really need or not that file, where you put. You just accumulate them because you have room. It doesn't matter whether you keep a lot of files that you won't need anymore. The same thing is for our memory. You keep everything, you just seem to get rid of it, of the memory, because normally we cannot retrieve it. In some circumstances we can. Again, it's been shown in mice, in old mice again. All mice forget. If you put an old mice in such a maze, they find a platform. If you keep challenging them, they remember it. Then, if you stop challenging them for a while, they forget it. And they start from scratch. Just because they're old, they forget what they've learned. Well, if you take an old mouse such as this, who has learned where the platform is and then it has forgotten it, and you treat it with anti-methylation drugs, he remembers. So it appears that DNA methylation is involved in making memories unretrievable, at least in old mice. Which means that memories are still there in the old mice. They simply are unable to find them. But enough of this digression. Let's go back to our main course. So, all the things, all the genetically modified mice I've shown you so far make me think that one day, perhaps, Icarus will need wax to put on wings. But then, there are contrary arguments. It is gorgeous to imagine that we can put into ourselves all those marvelous changes that I've shown. But then there are concerns, very serious concerns, such as untoward effects. How do we know that the modifications we introduce in our genome doesn't cause adverse effects? Well, we might know it from experiments in mice. Mice are not humans. Even if they are innocuous for the mice, that doesn't necessarily mean that they are for humans. We know that from pharmacology. We systematically experiments new drugs on animals, and many of them have now turned out not to be safe for humans once we transfer them into human products. So it is very difficult to make sure that a genetic modification is really safe. And again, as I said already a few times, safe means safe in the current environment. It is nearly impossible to predict what trade-offs might appear in a different environment that we may confront in the future. But you might say the same for natural selection. We are selected for living well in our current environment. We have modified our environment to live well in it. But on the other hand, we will be definitely off in other environments. All right, so even if we wanted to make sure that there are no side effects to the genetic modification introduced in the human during life, it would take a very long experiment with humans. It would take more than a generation. An untoward effect doesn't necessarily appear in the first generation of genetically modified humans or mice, for that matter. It might appear in the next generations. For technical biological reasons that I won't get into because it would be too long. So an experiment in humans, assuming it is ethically allowable, would take 100 years or more. Then, irreversibility of changes. Some of the changes I've described, such as eliminating senescent cells. Remember the senolytic drugs I talked about in the previous lesson, can be achieved with drugs. But drugs can be reversed. generally. You just stop taking the drug and go back to normal if you notice untoward effects. But genetic defects or genetic changes are not reversible. Once you have transmitted to your progeny, and they have other progeny and so on, if the gene takes a hold in the population, you can no longer get rid of it, unless you really go to draconian measures such as prohibiting people from reproducing or even killing. Also, who makes the decision? We would make a decision for our future fellow humans. They would have no choice. They would be born with the genetic manipulations. Whether they're good enough, we would know in advance. So who makes the decision? On the other hand, we continuously make decisions which will fall onto the future generations every day. So it's not so unheard of. Oh, and talking about the irreversibility of changes. Think of this other possibility. It has been shown that in some cases, a difference even in a single gene, can make two animals of the same species. This was done in Drosophilus, flies. Two animals of a single species unable to interbreed. That is, they are no longer fertile with each other. Although they are fertile if mated with other animals of with the same genetic modification. So, a genetic modification that was unfortunate enough to make newborn humans perhaps better, but unable to breed with the others, would in fact create a new species, a new human species. Now, think of a world even more complicated than our world is, in which we are all humans with the same genetic background, we are all the same species, we're in fact very, very similar to one another from a genetic point of view. You know about that? We are very similar to one another. Because many years ago, we barely escaped extinction. There was a point in history, about 150,000 years ago, when humans were a few thousands. Like many, many other Homo species that got extinct, we were on the verge of extinction ourselves. That's why we are so similar to one another. Because the genetic pool was so small that the differences were few as well. And yet, in this world of very similar humans, we regard each other as different. You're black, you're white, you're tall, you're African, you're Asiatic. Think of a world in which we have 2, 3, 5 different species that cannot interbreed. It will be us against them. It's a huge thing. Finally, changing human identity. What is human identity? I think we all identify ourselves with our mind. If we accidentally lose an arm, we are different, but we don't feel we are really a different person. But if you get Alzheimer's, then we perceive ourselves, another perceives perceive us as a different person. We are no longer the person we were. So, if we modify our brains the way we did with Google, we would still be human. We would be superhumans, or we would be just a different species. The that's food for thought. And so because of these contrary arguments, despite the appeal of the potential improvements I've talked about, it is because of these counter-arguments that we should think very, very carefully about modifying the human germ body. Especially, particularly, because it will quickly become irreversible. And that's because, and that's why, yeah.
I was wondering, this applies to us applying this on humans, but don't we, and I don't know how this sounds, but like always do science to perfect the mechanism itself outside of humans, so that our only restrictions end up being ethical? Because right now we're talking about irreversibility of changes and whether we might create an effect that we don't want. But if we manage to perfect it outside of humans, even in primates, for example, then the only question remaining argument would be the ethical one.
Yes and no. First of all, we don't do science to modify our environment or other animals or other plants. We do science to understand. Modification is technology, which in part stems from science. But it's different. Science understands. Technology does. Having said that, can we improve other parts of our environment? Can we improve ourselves? Yes, absolutely. Is there an ethical problem? I think there is an ethical problem. We will discuss it. Is there less of an ethical problems if we do that with cows, for example, or with I don't know, plants that we grow in our fields? Probably. We don't. Most of us don't feel there is an ethical problem in modifying plants. Some do. So, but you're right. It is definitely an ethical problem. But ethical problems are very difficult to handle. And so it comes to the point that even if we decide that we don't want to modify the human being, are we able to eternally prevent it from happening? Okay, here is something I wrote in an article in 2004. The history of mankind has shown that an acquired technology will not remain unused for long. On the contrary, someone, somewhere, will use it for one purpose or another, legally or against the law and the common good. Humans have not been deterred by the risk that their inventions and technologies might produce ecological disasters or mass destruction weapons. And they haven't really refrained, despite their solemn pledges, from colonizing the Antarctica or the outer space to gain power. And here's the main point. There is always a compelling reason, a national interest, a noble goal, or an irresistible social force that makes us open Pandora's box. So basically, what I was saying here is that once we know how to do something, sooner or later we will do it. Sooner rather than later. And so, what's new today? We are there. Less than 20 years from, far less than 20 years, as I assure you, after I wrote that. First there was a technological development, which is the technology widely known as CRISPR-Cas9. This is has been called sometimes the democratization of genetic manipulation. That is, it's a technology so simple that every decent biological laboratory can use it to manipulate genomes. It's easy, cheap, effective, and it works like this. If you want to introduce whatever change into a genome, you devise, first of all, a small guide RNA. An RNA which is a sequence identical to the place that you want to modify, or where you want to introduce a protein. Alright. Then you express an enzyme called Cas9. Many of these enzymes, in fact. The most commonly used is Cas9. Cas9 will cut the DNA where the gRNA anneals, and then you can introduce, by homologous recombination, as we've already seen, a modified version of that locus and obtain the modification you desire. Here I've shown a single permutation, but you can make larger and wider modifications. It's easy, it's effective, and it is relatively safe. Now, this as far as the technology goes. This has also got the Nobel Prize. In, what was that, 2015, I think? Yeah, in 2015, I got an email from a scientific publisher saying we have published a wonderful paper about this and that. I went, I followed the link, I went and read the paper. It was a CRISPR-Cas9 mediated gene editing in human triple-nucleozygotes. What does that mean? It means that they used the newly created CRISPR-Cas9 technology to modify a human zygote. Now, they use a triple-nucleozygote, that is, an oocyte fertilized by 2 sperms. So this is highly abnormal, and it has no chances of developing into a human being. That was to deflect any ethical questions that they might get. Right, but what's the purpose of such an experiment? It has only one purpose: modifying the human gene. You learn nothing else. And of course, it sparked an epic debate. People clearly understood that this was the premise to modifying the human genome, and debated about whether that should be allowed to be done or not. But eventually things got faster than the discussion, and and the technology even improved, so that now CRISPR-Cas9 has been modified so that it introduces fewer accidental mutations elsewhere in the genome, in addition to the one in 10. But anyway, it was just 2018, 3 years later, when this guy, a Chinese scientist called Wen Jui, announced that he had created transgenic humans. In fact, he had created 2 baby girls, 2 twins, in fact, from a Chinese couple. Now, why did he do that? That's very interesting. First of all, in China, as well as in many other countries, modifying the human germline is prohibited by the law. And in fact, he got first fired from his institution, and then he got jailed and spent 3 years in jail. He was released, I believe, last week. Second, there's the question of why he did it. he said, he still maintains, that he has done that in order to preserve the babies from disease. Because the father of the 2 girls was HIV positive. The mother was not. Now, the thing is, there was absolutely no need for this genetic modification. What he did was to knock out a receptor of GCC5, which is the natural receptor for most strains of HIV. So if you don't have that receptor, you cannot get infected by HIV. So Joey said, well, the father is HIV positive, so I want to preserve these girls from getting the disease. The problem is, first, that the mother was not positive, and the newborn gets the disease from the mother, not from the father. Also, it gets the disease when it is born through the natural birth canal, in the natural way. There is a very simple way to prevent it, that is cesarean section. So there was absolutely no need, but he argued, well, but he will live in an environment, in a household with a person who is HIV positive, their father, so they might get it from him accidentally. Well, it certainly doesn't justify modifying the human brain body. So he got jailed about that, but people kept speculating why, really, he had done that. First of all, it was to show, certainly, that was Hubbard's ambition, pure ambition, showing that you can do it. Showing the world that you are the first who have done it. But also there might have been other reasons. The same mutation has been introduced in mice, and surprisingly, it increases their memory. In fact, their cognition, it is their intelligence, by 40 to 60%. They are 40 to 60% smarter than normal mice. Now that's huge. If you are an average human, your IQ is 100 by definition. If you have an IQ of 160, you are a genius. So, it is possible that he really wanted to explore the possibility of making smarter humans. We don't know, because these baby girls are kept, of course, under strict medical observation. We don't know the results. The reason why we don't know the results is first because they are Chinese and the Chinese don't like to share results. Second, because it is ethically questionable to publish results that have been obtained in an unethical question. You know, the results of experiments performed in the concentration camps by the Nazis have not been published. They might have been user-free, we don't know, but they haven't been published because they were obtained by torturing people, and that's unacceptable. If we publish them, other people might think, well, it is worth torturing a few people in order to gain important knowledge. So we don't do it. So there is always a struggle between the possibilities, the aims, the potential, and the ethics that should guide our actions. And so we are at the end of the lesson, not at the end of time, because I spared time for you to debate this issue. Should we intentionally modify the human genome? And if so, what should be the limits? That's the question that we should ask. And I have an answer that I won't anticipate, of course. And I'll let you debate. Does anybody want to share some thoughts about this?
The only thing I would say is that, in my opinion, anything that touches the germline is very iffy. Should be considered anyways. Somatic mutations, okay, but like anything that touches the germline is always vacant.
Do you all agree?
So I do not honestly know, because is just the potential for danger our reason to stop exploring something? That
Excellent
presents a
question.
huge block in evolution.
But we're forcing evolution. It's not evolution.
It's artificial.
We forced so many things and it worked for us.
We are past evolution.
Why should
Like we already started to I think we already started to go past the evolution. Like I do remember that we were talking about the BRCA gene and when the BRCA gene that increases the risk of cancer, and Angelina Jolie removed her breast, and that was opening the door for, I don't want to call it eugenics, but somehow, I don't know. I feel like
I would call it
like,
prevention, but it's okay. It's a matter of, It's a matter of
No,
debate.
No, but no, sure, but still, you do that for cancer, and then at some point you will maybe do it for other things. And I like when you wrote hitting the head in the sand. I don't think there's a way to stop this somehow. I feel like there are dynamics in the world that just happen, and we don't want the Chinese to do nothing before us. I don't know. Like, no, I feel like this is exactly what is going to happen. Like, who knows what they're doing? So
Whether we
better,
like it or not.
yeah, better we do it too. And I don't know. I feel like this is so better to talk and actually put limits and ethical. Although that will open like huge debate, of course, and that's right. I mean, it's okay. But
I also really like the part of the speech, because I think it's exactly what you said. You cannot prevent something so interesting, something with so much potential to ever be explored, so it's obviously better to do it in the open. But then another argument I wanted to say is, we don't find ethical problems with, okay, maybe not find ethical problems is an overstatement, but it is allowed to have an abortion because you know that the genetic problem defect will present itself after birth. So if that is allowed, you supposedly, and something that many people argue, stop a life because you know it won't have the potential to have the most comfortable life or the longest one, etc. Why would you not explore the option of, instead of just stopping that life, try to make it better, or try to prevent that problem? I mean, I feel like it falls very close to that argument to present it as if it's a completely different conversation.
I agree with you, and the answer that some people would give, I don't buy that answer, but the one answer some people would give is that in the case of abortion, we are dealing with natural, unwanted natural mutations. So we are preventing damage. In the case of intentional genetic manipulation, we are potentially causing damage intentionally. So it's different.
But
Yes,
if
but
we're
it-
correcting a mutation, it's still.
But that's still not allowed, just correcting the mutation.
Well, no, in fact, it is interesting, in the last few years, the debate turned from modifying the germline should never be allowed into up to what point and in what circumstances should we modify the human germline. So it has already shifted quite a lot in a few years, just because it became possible. Previously it was a nearly theoretical possibility. It was, before CRISPR-Cas9, it was at least extremely difficult to do. With CRISPR-Cas9, we are navigating up to what extent.
And in the case of modifying the germline, how do we know that, let's say, the infant that resulted after the modification is the same infant, is the same human that would have existed without this modification?
You're opening
Of course, we don't know.
philosophy question.
Ethics.
Well, ethics is philosophy.
I'm really frustrated.
You first.
If you were to transfect genes into the germline, how do you control for gene expression in every cell of the body throughout the life of that individual? Like that you want this to express in specific genes, or do you allow for the mechanisms already in the cell to allow for that?
Yeah, we can do both things. We can either choose a mechanism that we like. For example, overexpressing something, overexpressing means expressing at higher levels than normal in this or that tissue or anywhere in the body, everywhere in the body, using appropriate promoters for the gene. Or, alternatively, we can just copy what nature has already made, that is, introduce the whole genetic locus of that gene, and so let it be expressed in a physiological fashion, depending on what we want to achieve. We are good at doing both things.
I find it very frustrating because I feel like even if there is the potential of pushing the limits a bit more towards the advantage of humanity itself, it still reaches a dead end at some point, because the argument you said before about creating a new species and creating even further diversity seems inevitable if we start exploring that. And I can't even imagine how things would work after that. In that sense, I feel like it's either hitting a dead end or a huge difference in how we perceive everything. But I want to hear, what is your take on this?
What is my what?
Your take, and the limits, should we modify, and the question itself.
All right. Let's disclose my. Yeah,
One
please.
question
was
before
rushing.
before you give your
Everybody else before me. Yes.
Like as you give your answer, how long do you think it would take if someone is able to introduce a mutation that gives an advantage, but also like is just on the tipping point of creating a new species? How long would it take for this there's actually a separation between species would be?
If we did create a different species, meaning
Yeah,
a
they would
species
have
that cannot interbreed with us, would be instantaneous. The first baby would be in such a
Because then
position.
it would have to be, we would have to have at least 2 babies so that they could.
Yeah, otherwise it would get extinct.
Yeah,
Yes.
okay.
So can we put the limits on based on equity rather than equality? Like if a couple is having a genetic condition and obviously they know the baby which is going to be born will be severely affected. In that case, I think it makes sense to modify the germline just to give an equal chance like the rest of the normal babies.
But then that creates a lot of problems, because if you're not just talking about life-threatening disease and just cognitive function one, you are saying that that child itself, because of having lower cognitive functions, should be genetically modified to have better ones, and then another couple will come and argue that they have an average lower IQ. Should their kid have the average IQ of a human, or should they have a lower IQ? I mean, I feel like it opens a huge debate there if you just try to create an equity.
Yeah, probably what you said before is the same thing. You know, how do you draw the line of what really affects your life enough or not? You're gonna fall into that, but just gonna happen.
And if you raise the bar, like if these girls come out as 160, then you're really like people will be who are even 90 IQ, which is a little bit lower the average, but still normal, will be at that point then pathological compared to the other.
So I think, for example, one thing could be that maybe a condition is a problem, but not a life-threatening problem, but let's say that in a country it happens, for example, it's overrepresented, so maybe it causes a problem for, I don't know, the NHS or competition. And maybe in other countries have less of that. So maybe countries could, put looser rules on that, and the other one not, because it's not such a huge, I don't know how to call it, like administration or political problem for them. It's never ending.
You should have like a world committee, like that, I don't know, like something similar to nuclear weapons, I don't know, like something like that is checked.
It's something that's never existed.
Oh.
We have the United Nations, but you see how poor their performance is. So we don't have such a body for one thing. Second, even if we had it and we decided not to do something, somebody like each and you will do it anyway, illegally. Who cares? Because it's so easy and it can be done.
On the other hand, just abolishing the potential of exploring something so big seems so unfair. I don't know who, or the science itself.
Okay,
but if-
it's the second time you raised this point, and I've taken it already, and I have not an answer to that, but I have some slides that I brought just in case this point came up. So I'll show you them in a second. So, if you want my own personal opinion, which counts for one, so it counts nearly nothing, is that, first of all, changes in the human germline are essentially unavoidable for the reasons we have just said. Nobody can say that. If they are, I agree that we should at least guide them as best as we can in a good direction, whatever is a good direction. But I, frankly, honestly, I see the risks, I see the dangers, but I love the potentialities. Oh, yeah.
And then like another argument that you came up with. It could be argued that the previous generation and our generation are creating a world full of more carcinogens. Wouldn't it be, which by principle should be something unethical, wouldn't it be ethically fair to try and do something to prevent the problems, the things you caused?
It's an excellent point. I agree with you. We are polluting our world, we are making it carcinogenic, and we should do something about it.
Exactly, like the argument is you're not allowing...
Such as not polluting, but
Yeah.
we don't seem to be very effective at it.
I mean that the argument people are trying to use is that you're not allowing nature's natural evolution of things, but we haven't allowed it in everything else. Aren't we supposed to account for that change? Or let some things run naturally and everything else we have a say on? You can...
I'm not sure I understand.
Let me rephrase because I really lost it there. The point was that we are creating a world which we alter extremely with what we do, and the argument to not touching the human genome is you're not allowing nature's evolution and nature's natural selection to happen. But we're not allowing nature's course in everything else. Why should we allow it only for the human evolution? Shouldn't it be taken into account that everything else is changing, so we should do something to follow that change?
So basically you're saying, why are we special? We are changing everything else, why not ourselves?
Why are we not protecting
It's a
everything else, saying we're doing it for the better of everything, but not here?
The answer would be just because we are us. We care about ourselves more than anything else in the world.
But is it really caring about ourselves, or is it just having tunnel vision and we don't touch?
No, maybe the answer you said, like, there is the answer. Like, what if the earth becomes unsustainable in the future? We need to do something to ourselves, modify ourselves, so that we can adapt to the world, the future world, which becomes unsustainable due to lack of oxygen, water, and other.
But it's a slippery road. So we destroy our world and we make ourselves capable of standing or withstanding what we have done, and then we make it even worse because now we can withstand it. It's an argument, I'm not saying it's wrong.
Even there, like, how do you establish what's the cutoff point? No, like, when do you, like, everyone has to start to, heavily modify or to that? Yes, of course, it's not going to be a thing that you can start, slowly, but it needs to be massified. So we should, like, have a definition of the end of time.
Also think we're saying this and we're approaching this as, oh, everything is going to go fine, everything is going to go well, we're going to be enhanced and everything. But our limitations is really the knowledge of what we know of biology and our brain works and our, we showed before the interconnectome, and we saw like every protein is connected to all these other proteins, and each cell has this network. So you're saying, oh, we alter one part of the network, but what happens to the remaining part of the network? So there are really sometimes unpredictable things that can occur. So what if we kind of screw up and we create something that can potentially go in the opposite direction of what we wanted? So instead of making something better, now we're doing something worse, so we can drive ourselves to extinction because we were too ambitious in air quotes.
I mean, that is undeniably a very
Different.
I'm sorry.
I was gonna say, yes, you have a point, but like when science start, like the amazing discoveries will come by mistake. You do something, you see consequences. I mean, it is humans we're talking about. You do something, you change, you see the connections, the communications. If you insert like a protein that is not human, not genome, we don't know how this is gonna interact with all the other proteins in our body. It might be beneficial, it might be bad, it might kill us immediately, we don't know. We might not try it.
But even a step before just trying it, I was saying, like before we started the whole debate, that even since we're at the point of discussing whether it should be allowed or not in humans, before we even start talking about that, shouldn't we have reached the point of perfecting it or have run the most data checks and the researches we could have done in anything else? Like, for example, primates, the closest thing we could test in humans.
I don't think that's possible. Usually research doesn't work that way. It's, you go for the more empirical, especially when you don't know the subject very well, and you see how that happens. If you try to overthink, it's going to take far too long.
But that's what I'm saying.
Far,
Empirical
far,
tests,
far, far, far.
not sit down and
But then the problem is like,
The way
we live in a
technology
world
works is we try things,
yeah.
they work to some extent, and then we improve on the first tries
Yeah.
progressively. Now, if that involves living beings, especially human beings, it takes a toll. So, please.
No, I just wanted to say that we're gonna at some point choose a cutoff line, and it's an illusion thinking that we can choose the right one. Like every choice we will make will have pros and cons.
Plus
I
and
think.
minuses. Yes.
And probably we will see the results only after. And like many things in history, like we don't understand anything about the present, and we'll probably the best will be just be cautious and do like really small steps, and preventing it to skyrocketing to something that we can't control anymore. But it will be an illusion that we do that and then we'll all be happy about it, or there won't be problems. For sure there will be problems. Problems comes up from.
Absolutely.
So,
I already mentioned what happens with drugs. We do our best to predict what drugs will work and will be safe in humans by experimenting first in the laboratory in animals, but then most of the drugs, once they are tried in humans, fail. The vast majority of them fail, despite performing well in animals. So it is simply impossible at the present state to be sure, or even close to sure on that. And so, going back to, you said two interesting things. One accidental, the second intentional. Both of them are. By the way, one observation. Last year, most people in the class were strongly in favor of modifications. They said, if we can prevent humans from getting tumors, let's do it, period. It was very difficult to show them that there were counter-arguments for that. And it is understandable. It is understandable that in the face of the immense burden of suffering, of death that tumors provoke, not to speak about other diseases, it makes sense that we try to prevent that, even at a cost. Everything we've done so far has had a cost. We have explored the world at the cost of the lives of innumerable explorers. But that's the way we work. We put our lives at risk when we wish to know something. Okay, here is what I wanted to show you. You see, you mentioned that knowledge is a good thing in itself. And in fact, we have long thought so. The knowledge couldn't be evil. Knowledge is always good. In fact, our Constitution says art and science are free, and free is their teaching. Period. There are no caveats, no qualifications. Art and knowledge are just good. Now, I'm afraid, though, that we have become so powerful, technologically speaking. Some domains of our knowledge have such a potential that we are on the verge of not controlling the outcomes that stem from our knowledge, the technology that our knowledge made possible. And so what you asked, should we not explore some domains of knowledge because they are too dangerous? You said that as a paradox. Can you possibly imagine of not exploring some parts of the world just because it is too dangerous to venture into that? I think this would be theoretically the only possibility to save ourselves. Never to explore domains that are too dangerous to gain knowledge about. But I don't think it is possible. For a variety of reasons. One is that there is no universal body that makes decisions about that. Second, because you cannot judge about the desirability or the dangers of the knowledge domain until we have gained that knowledge. And once we have, that's it. I'd like to show one example. By the way, artificial intelligence right now is an extremely serious problem. Or at least many people think so, and I do too.
You said problem?
Sorry?
Extremely serious problem?
Yeah.
Danger.
I accept one of the two terms. I feel like problem is a big umbrella term. It doesn't allow the possibility of advantages. Whereas danger, I cannot deny.
Okay, let's say a serious one.
Yes.
I accept your correction.
No, it wasn't mine.
But I think we are giving much less importance to artificial intelligence, for example. Not much discussion about it,
No.
about the danger and eventually the ethical problems which
Not
might
nearly.
not at all compared to what we are discussing today.
Exactly.
So and that danger is not felt at all in the general opinion, I would say.
In the general public, it is not fair.
I feel like it,
Among the specialists, they do know there is a danger.
Yeah.
But basically, there is also a race. And when there is a race, nobody cares about dangers. If for no other reason, because everybody thinks, I see the danger, but if I stop, they will continue.
Yeah,
So I better continue myself and get there before them. So we'll never stop.
I feel like it's harder to have conversation about subjects, especially in the general population, you understand less. And I feel like artificial intelligence falls in that category for sure. That an average of people have a very little understanding of what we're talking about. Whereas if you try to have an argument, a debate about modifying a human genome, at least some basics you have fundamentally set.
Or it's just that we are dealing with a human, and so
And
there's
you
a
just
lot of
talk about
ethical
that.
going
Yeah,
on.
that's it.
Whereas computers are just computers, and
No, exactly.
there's no obviously unified
I'll
system.
address that in a minute. It's very interesting. I'll be short because we are at the end of our time. I'll show this geoengineering. You know what geoengineering is? Is a class of technologies that want to modify the whole planet in order to make it better, such as sending something in the troposphere to reflect some of the solar light in order to cool down the planet, or seeding iron in the oceans in order to increase the production of plankton, which will feed whales and other fish species, in order to increase the number of these species in the oceans, and so on. Now, on this scale, these technologies are so dangerous, and there will be no coming back if we ever implement something like this. We could be in a catastrophic situation. Even we could extinct ourselves. We get ourselves extinct. So, this is the first time I see in a scientific, in a prominent, in a top scientific journal, saying that no, it's not here, here. Arguing that we should not even explore these possibilities, because once we know something, once we develop technology for doing something, we will do it. So in a scientific journal, they say, this is something we don't want to learn about.
I don't understand why.
Well, look,
Because once you throw something in the atmosphere and it reflects more light than you suppose, and you freeze the whole earth, the whole earth, there is no coming back.
Yeah, but you also have to deal with the problem that you literally have in front of you. But you also have to deal with the problem that is literally in front of you, global warming.
Yeah.
So how is it?
Do it the hard way.
Yeah.
Produce less CO2. Not the apparently easy way. It is putting reflectors in the atmosphere.
But do you have to throw realism in the conversation as well? What is realistic to happen and what's not?
It is realistic. It has been calculated and demonstrated that it is realistic. It is possible to reduce.
I don't doubt that. I'm saying, is it realistic that things can change this fundamentally where so many big forces are
Yeah. It has been done on a small scale and it works.
I'm saying if it's
Is
possible to
considering
be implemented.
human beings
Yes.
and their minds.
Exactly.
Would they
Not that
accept to do that?
Yeah, not that
That's
it's
what you were-
physically possible.
Yeah.
You think so?
Tell people that they can keep living the way we do. We can waste things, we can produce as much CO2 as we want, because we are throwing something in the atmosphere that will take care of the problem. People will accept it.
And now that we've told them that you have started the clock running backwards, and when the clock ends, we probably are reaching an irreversible point, what has changed? Minor things.
Sorry, leave me.
I'm saying that even now that we have presented to the whole world, fighting it for years, that global warming is a problem and that it will reach a point, if it hasn't already, of irreversibility.
Oh, I see. I'm not that pessimistic. I think we have shown that we can do something about it. We aren't doing it nearly as fast as we should, but there is clearly a worldwide consensus that we should do it and that we will do it. The question is whether we will do it fast enough, and that's a big question.
Exactly, but when it's just a question, so we're playing a lottery, whether we'll manage to do it and keep ourselves into following some rules or change and implement some changes. Should we not also explore other options since that one is not working as we expected?
Yours is a perfectly acceptable point of view. In fact, in these other editorial measure, the editorialist argues that if solar geoengineering is harmful, leaders will need evidence, that is knowledge, if they can rule out the technology. So that's exactly what you're saying. So, and there was one last point that I think again you raised, and what was it? I said I will address that. You also, what did you say? What did you say last? You said too many things to remember.
The last thing you said?
Yeah, what did you say last? What are you talking about? I know it's not easy. She doesn't know.
I don't remember. Something about the cutoff line? Or, oh no, that people are not are giving less much attention to artificial intelligence because it's just about computers.
Oh,
and
yeah, good. All right. So, very quick and last thing. So, because you called for it. It was my fault that I mentioned artificial intelligence. Now let's say a few words about it. You both said, I think, that people don't really understand artificial intelligence. How can they make a decision about it? How can they perceive the dangers of it? Well, they do. You know why? Because everybody has read science fiction books or has seen science fiction movies. Science fiction is extremely interesting because it explores in advance possible future realities. And it makes you think. The best science fiction makes you think about the possibilities. In general, I think, that's my own viewpoint. In general, literature really, literally guides science. That is, scientists work on what they think it is explorable next. They don't look much farther away. What tells them in what direction they should move is the literature. It wasn't scientists who thought for the first time to go to the moon. It was poets, it was artists. Then came the scientists and they said, oh wow, this is really a great idea. And so let's see if we can do something about it. So, even the general public understands the potential. Everybody has seen Terminator. If you make machines that make themselves better and better, and then build other machines, and we are there already, you get a Terminator type of scenario. And people understand that. They simply don't know that we are very close to that position. Very, very quickly. What were you saying? Here, just in the last few years. And this is what published. Of course, the unpublished things are much further ahead. of what we already know. Here, this is an algorithm that plays chess, shaggy, and go. 3 entirely different games. Better than human champions, world champions. And you know what's the output of this artificial intelligence platform? The rules of the games. It has got no other input, not output, input. All the platform was given was the rules of the games. It is as if I gave you the rules of chess. You thought about it, and you became the world champion. And that's exactly what this platform does. It has played hundreds of thousands of games against itself, and it has learned the rules and immediately beaten the world champions. Here. Why is it not done? Okay. And now look at this. This is amazing. This is another artificial intelligence platform that designs computer chips, CPUs, the core of the computers, the real computing part. The rest is accessories. Now, it designs that. It takes human engineers months. They work in teams, not one by one. Human engineers work in teams, and in several months they can design a new chip. This platform takes hours and makes chips that are at least as good as the ones we design, and improves itself every time. It learns from what it has done and makes it better. So now we have a computer that is capable of making other computers. And computers can learn the rules of chess and beat all human beings. Do I have this other one? Yes. This other artificial intelligence platform makes code, computer code. Well, it's just a rough, first draft. It makes computer code as a junior human technologist. But there is ample margin of improvement, they say. In a short while, we will become much better than humans at making code. So, if we put these things together, we have artificial intelligence that learns on its own without human input, that can build other computers, that can code, that can make code for those computers. We put 2 wheels and 2 arms on that, and it will eat you. It doesn't need you anymore. So, this is another factor. It's not biological, it's not medical, so it doesn't concern me.
It does actually concern us because this